but I do want you to be familiar with all of this material. So we're looking at how we now, in the 21st century, actually examine synapses and how we can actually visualize synapses. I had mentioned and started to give you a little bit of what Carl Dijsselroth had done in terms of optogenetics, which will be the major focus of what we're going to be talking about today. But I also have to tell you a little bit about the team that he assembled. Like, all of these different individuals were really, really young. You can see that they're all now assistant professors or associate professors at different universities. But he actually assembled a group of really, really young individuals to tackle things from a perspective that no one had used before. And so people like Ed Boyden, for example, Fang Zhang, and Viviana Gradinaru, again, the names not being important, all of them actually created this cohort that really started to think outside of the box. And they not only came up with optogenetics, which I'll be talking to you about in the next few moments, but they also came up with a way to actually make the brain clear, which is known as clarity, another really big innovation from their lab. And this is the way that they think differently. And again, you don't have to worry about these details. But if you're looking at how to look at cells, and cells are incredibly small. They're on the order of hundreds of microns to tens of microns in size. One of the ways that most people would tackle this is to build better, bigger and better microscopes. But Ed Boyden, who's here in the middle, actually decides that he wants to take the opposite approach. So instead of building a more powerful microscope, why not try to take something really small and actually make it bigger? And that's the way that this group thinks. They think really differently than most other groups do. So he's actually developed a new technology which is known as expansion microscopy. And expansion microscopy allows you to take small cells and then expand them to 100 or 1,000 times their normal size. And of course, you all know Fang Zhang, who's been doing work with CRISPR-Cas9. But this is one of the beauties of getting young people together to tackle problems that have been plaguing neuroscience for over 100 years. And this is sort of the solution that Stanford has come up with. So I'm going to go over in detail how we use optogenetics to understand neuron function and how we understand the release of neurotransmitter at the level of these different synapses. So this is actually not trivial. It actually involves a number of different things where you have to plan your experiments, which is some of what you're going to need to do on that pink, purple sheet, and then to coordinate these different types of experiments. I don't want to give you just a glossed over version of what optogenetics is, because many of you going into fourth year courses will hear about it, and I need you to be really critical thinkers, just as Carl Dijsselroth has assembled. So this also means that we have to talk a little bit about Cre driver lines, and I'm going to talk to you about an enzyme known as Cre recombinase that some of you might have heard about in your first or second year biology classes. And then I'm going to talk to you a little bit about why we need to do these things in the first place. Why bother with all of this? Isn't everything that we've looked at in this course so far in examining a synapse, isn't that good enough? Why do we need this extra level of technology? And then at the very end, I'm going to talk to you about chemogenetics, or the older term is known as pharmacogenetics. The new term now that is used by neurobiologists is chemogenetics. And I'll talk to you a little bit about this. If you understand everything that I talk to you about optogenetics, you'll understand everything about pharmacogenetics at the same time. In today's lecture, as we go through them, again, just to introduce another wrinkle to this, I want you to focus your time and how you study really well. And I want you to make sure that some things I need you to know. And again, this is Russian, and this is this Cyrillic? So, Max, I'm going to ask you, if you're saying something yes, how do we say this in Russian? Da. Okay. Or if we're saying no, you don't need to know this in red, this is нет. Okay. So, I'm actually going to start off by telling you something that you probably have heard about in psychology. And of course, you don't actually have to know the story behind this, but it's actually one of those interesting preludes to optogenetics. This happened in the 1950s. And in the 1950s at McGill University, there were, again, these really two young investigators who wanted to tackle one of the biggest problems that was plaguing neuroscience in the 1950s, which was, how do animals stay awake? And one of the things that they were trying to do was to implant an electrode. And on the left, you can actually see that by the 1950s, Skinner had developed his Skinner box, so people knew how to implant electrodes. They knew how to do self-simulation studies. And James Old and Peter Milner at McGill University actually tried to do experiments related to different areas in the brain by implanting an electrode in the brain and turning on the electrical currents to see if they could figure out what areas of the brain were responsible for keeping animals awake. And one of the things that happened in this particular experiment, which is what you are seeing right here in the middle, when they turned on the electrode, they were actually observing a really unique behavior in these rats. And these rats that had this electrode, where they originally planned to stimulate these rats to keep them awake or put them to sleep, actually started to move around the box. Whenever they stimulated a rat in a particular area of the box, the rat would go back to that area of the box. When they turned off the current, the rat would move around freely, but when they turned the current back on, the rat went back to exactly the same place over and over again. This one rat became the most important rat in the history of McGill University. In fact, this is the only rat because they couldn't reproduce this in any of the other animals. They only saw this in one animal. None of the other electrodes, when they stimulated, caused the same behavior. And one of the things that they realized is there's something really unique and something really different about our experiment here. We need to take an X-ray. And this is an example of the X-ray that they took. You can see this is where the stimulating electrode was placed. Can you come up with an explanation as to why this rat would move to the same area of the box whenever an electric current was applied to it? Remember, they're looking for the brain area that's involved in keeping the rat awake or having it go to sleep. What happened? How would you describe that? How would you come up with a theory to try to explain this? They tried for a while to try to explain this entire phenomenon. And they came up with this idea that maybe they had stimulated an area of the brain that led to curiosity. It turned out much later that they had stimulated an area of the brain that we're going to be looking at today, which is called the medial forebrain bundle, which connects two areas of the brain, the VTA and the nucleus accumbens. What they were doing is really releasing a lot of dopamine when they were stimulating that electrode. So whenever the rat was in a particular place, there was a flood of dopamine coming out saying that this is a behavior that should be rewarded, in other words. But they thought that they had actually found the area of the brain that was related to curiosity. That was their explanation for this. So this becomes a really big issue in neuroscience. And for the longest time, up until the year 2006, when the very first report came out, when we were looking at problems like this, trying to figure out what area of the brain does something, we would do something like this. We would stick in an electrode. Again, it's really, really small. I don't know if 1.27 diameters and millimeters in diameter seems like a really big electrode to you, but it's very, very small. It's literally the diameter of a wire. And then we were trying to stimulate specific targeted neurons. But here's the problem. This electrode actually generates an electrical field, which is shown here in pink. Around the outside of it. And one of the things that happens is, even though we are hoping to stimulate only the neurons in this area here, we're actually stimulating a number of different areas. Now, Peter Milner and James Old actually missed their target, which is why they ended up getting the effects that they did. But in other instances, when we are on target, when we hit the appropriate groups of neurons, we can never say with any certainty that we are only stimulating neuron A when we see other neurons in the area. These adjacent, non-targeted neurons that are around the same area are also going to be impacted by this electrical field. So we were always dogged by this inability for us to say with 100% certainty that we're only stimulating the neurons that we're hoping to stimulate. And this became this big problem. So we've, in the past, always stimulated other groups of neurons through this electrical activation. It's also true that when you're looking at cognitive psychology, one of the big things that happens in cognitive psychology is we talk about lesion studies. You lesion this area of the brain, you see what the resulting behavior is, and you make the equation that if you take this part of the brain out, and then you look at the resulting behavior, that part that you took out must be responsible for a missing behavior or an added behavior. But again, there's no way for us to tell with any certainty, even if we're the best neurosurgeon out there, that we aren't actually bringing out or lesioning other groups of neurons. This level of precision, in other words, actually is not sufficient for us to tell anyone that it's only neurons A, we are not stimulating neurons B and C, even though they are found really nearby. There's also this other issue. If you're doing lesion studies, there's no way for you to look at the temporal effects. You can't look to see what happens over time. And so controlling things temporally after a lesion study, as well as the specificity, is lost in many of the ways in which classical neuroscience has approached these different issues. So was there a way, in other words, and this was the problem that was presented to Karl Diceroth, can you take care of this issue? No one believes with any certainty that when you stick in an electrode or you take out a group of neurons, that it's specific to that area. Can you show us in some way that you can control the temporal firing of the group of neurons that you are specifically trying to demonstrate is responsible for a behavior? And again, as Karl Diceroth is a clinical psychiatrist as well as a bioengineer, he approached this sort of in a very different way. So again, these are all the things that Karl Diceroth was faced with. There's poor spatial resolution. We can't identify if we're stimulating groups of neurons or indeed the neurons that we specifically want to be able to stimulate. So there's that confound. Is it perhaps other neurons that we're also stimulating? The drugs that were available in the 1990s and the early 2000s, even though we had blockers, and we've talked about several different blockers in this course already, even though these blockers existed and that were relatively specific for groups of neurons, we were never able to demonstrate, because of the time it takes for these drugs to work, that the pharmacology being over 30 minutes, that indeed the neurons were directly being affected. And again, for lesions, once you take them out, you can't actually show a time course once those neurons are gone. So these are all of the different issues that Karl Diceroth is left with. And how does he decide to solve this? If we were taking a traditional approach, just like we would with a microscope, we try to build a smaller and smaller electrode, or we try to build electrodes that were so small that they would only stimulate like 1 or 2 neurons at a time. Many scientists had been trying to do that unconvincingly before Karl Diceroth actually tried to do so. So he decided to take a very different approach. Your brain, the brains of mammals, are encased within a skull. That skull prevents light from getting into the brain, and as a result, your neurons are continually bathed in darkness. There is no light that is hitting these neurons. He decided to take a look in terms of the genetics for any types of channels that might be activated by light. If light can't enter your skull, and he can actually introduce light, could he then use this to his advantage? So he starts looking at other types of channels. And as he starts looking at the genomes of other types of organisms, he realizes really quickly that chlamydomonas, for example, a type of algae, actually has different channels that are light sensitive. Now, these are non-specific cation channels, and I'm going to repeat this again a little bit later on. And these respond incredibly quickly. That's the other thing that's important. As soon as you turn on a light, these channels have to be activated just like your neurons are activated when the membrane potential changes. It has to be just as fast as being able to generate an action potential. And he was able to show that these are responsive on the order of milliseconds. Is that close to what we see in the nervous system? Do we get millisecond activation of channels? Indeed it is. So once you activate a channel, a voltage-gated sodium channel, a voltage-gated potassium channel, a voltage-gated calcium channel, as soon as it detects a change in voltage, it responds on the order of 1 to 2 milliseconds. You see an action potential, in other words, on the order of milliseconds. So again, there's a lot of controversy about this recently. Was Karl Diceroth, was Ed Boyden, was Feng Zhang, and others, were they actually the first to come up with optogenetics? And there's a lot of controversy as to whether or not a lesser-known scientist, Dr. Pan, had actually submitted his abstract and his paper to the journal Science before Diceroth's group were actually able to publish. Many researchers were involved in this initial publication, including Gottschalk as well as Ed Nagel and others. But this again has become a really big area of interest. Who was actually the first to come up with optogenetics and this whole idea? Was it in fact Dr. Pan, or was it actually a combination of other people coming up with the same idea at the same time? So I'm going to tell you that this last molecule that we looked at in the last lecture, from the BTA to the nucleus accumbens, I had talked to you about channel rhodopsin. That is not something we normally find within the mammalian nervous system. That is something that Carl Diceroth and others have genetically engineered. We have to introduce that gene into our neurons. These channel rhodopsins are a group of different proteins that are channels that once activated by a specific wavelength of light, and here you can see the fiber optic cable, which is known as an optrode, going in to the brain of an animal at a specific location, that when these neurons that are now expressing channel rhodopsin are activated by blue light, those groups of neurons will become activated, and we say that channel rhodopsins are generally excitatory. Now, I said that you don't need to know this right now because I'm going to repeat the same thing to you in a few moments. So turning on neurons is incredibly important. Turning on neurons specifically and over a very short time window is incredibly important. But equally important is our ability to be able to turn these neurons off. And halorhodopsins can do the same thing. These are generally inhibitory. We need to express these as genes. We normally don't find them in the mammalian nervous system, and these are generally inhibitory when they are expressed in neurons. We do not use the same color of light. We activate them through either yellow or orange wavelengths of light, and when those neurons see flashes of yellow or orange, those neurons that have halorhodopsins will stop their activity. They will become inhibited. Is that okay for everyone? Do you have any questions on any of this before we go on? Have any of you seen a live demonstration of optogenetics where you have seen an animal with an optrode or a fiber optic cable where you've seen little flashes of light changing the behavior of an animal? Okay, so again, Armin works in a lab that works on channel rhodopsins, but it is an incredibly powerful technique that's used here at U of T. So again, I don't need you to know all of the details on this particular slide. We don't have these opsins. These opsins don't exist in the mammalian genome at all. These are foreign genes that we have to introduce into these neurons. These are normally expressed by microorganisms like Chlamydomonas, specifically algae. And again, we use transgenic methods. We have to introduce somehow, and that is the topic of today's lecture, this opsin gene that responds to different wavelengths of light into the genome or into groups of cells within an experimental animal. It is very hard for you to inject DNA or a gene, a naked DNA or a naked gene, into a cell or a neuron and to have that gene being expressed. We have to use other techniques to package the DNA and package the gene to have it expressed within these neurons. And this often means that we have to talk about viruses in the delivery of this genetic construct. Now keep that in mind for those of you who don't have a biology background or if it's been a few years, that we need to package this opsin gene into a particular virus that I'll be talking to you about later. So importantly, only certain cells will actually have the machinery necessary that's necessary to express this optin gene. We can control which cells will turn on this opsin gene if it's being expressed or not. And this comes back into the level of specificity that I left off with at the end of the last lecture. And this is an important aspect of what Carl Diceroth's group did. Again, any questions before I go on? Yes, Luke.
Why do the light channels must be fast enough to catch up with action potential firing?
I don't need it to catch up to action potential firing. I need it to actually take the place of action potential firing. I need it to follow the pattern that I want to introduce, and it has to be able to produce action potentials. Every time I flash on a light, it has to produce an action potential. And those flashes of light are very, very short. They're not like continuous. We can continually stimulate an area and keep the light on. That also gives us useful information. But for the most part, we flash on the light for 1 or 2 milliseconds, then we turn it off. So obviously that's much faster than you can control by hitting a switch, and so we control that using a computer controller. Any other questions? Yes.
I was just curious, can they put like these options in specific neurons?
Yes,
Go into detail?
so we are going to go into detail how we get these different options into specific groups of neurons. If you're looking through that purple sheet, that is one of the questions that I ask you. How do we make sure that only some neurons actually express, specific neurons express these different options? And I'll get to that. You need to understand that by the end of today's lecture. So these are sort of the first series of experiments that Carl Dijsselrath's group were able to report. Now these different types of algae, Chlamydomonas reinhardtii being one of the most common types, actually express this little sensor. And what you're seeing here is a petri dish that is full of algae in green. And as we shine light in one particular corner, all of the algae are migrating to the corner in which the light is being shone. In other words, they have a way in which they can detect where light is, and in response to light, and this is very slow, this is over the course of several minutes. By 6 minutes, you can see that most of the algae are over here. They are expressing the native form, what we find in nature in the algae, known as CW2. This will be mutated using molecular biological techniques by Carl Dijsselrath's group to allow this to be much faster. But you can see that we already have mutations where, for example, in response to white light, these algae, if they have a particular isoform, actually are no longer responsive. Even though the light is being shone with a particular type of this channel rhodopsin, they don't move. If you can't detect it, it doesn't cause the movement of these algae. And again, once they were able to clone this and they were able to purify it and they were able to actually change the kinetics, they were able to create what is now known as a channel rhodopsin. Every time you flash on a blue light, this neuron that expresses it, that has this channel, now will open and then maintain a steady state and close as soon as the light is turned off. Again, if you turn it on again, it opens, stays at a steady state, and then closes as soon as the light is turned off. So in other words, it matches the time scale of how we flash on and off the blue light. There's no lag. It's not like if we had a lag, if we turned on the blue light, then we had to wait for the channel to open. It wouldn't work very well for us, but there is no lag. As soon as we turn it on, the channel opens. As soon as we turn it off, the channel closes immediately. And again, one of the things to make sure that you are aware of, what makes all of this work is very much like the retinal that all of you have. Where do you have this protein known as retinal? In your eye. Okay, where in your eye? Okay, in your retina. Where in your retina? In the rods. Okay, and you know that these different types of proteins, like retinal, have different conformations. They either have cis or trans. In this case, instead of being activating a G protein, what will happen is that in the channel rhodopsin, this molecule that looks like retinal, shown here as a key-like symbol, in response to blue light will actually change its conformation, allowing it to open, and then it is a non-specific cation channel. Okay, so it only opens when light hits it and activates it and changes its conformation. And then you can see that both sodium and calcium flow in, and then potassium will leave. All of these events causing depolarization in the neurons that actually express this channel rhodopsin. So this whole molecule is channel rhodopsin, and the part that is actually opening and closing in response to light resembles retinal that you are seeing here, changing its shape in response to light. Any questions on that before we go on? Yes?
The CW2 so that is expressed normally in nature. What is it exactly? Is it a type of channel or is it
It's normally not a channel. It's almost like an eye. So yes, I know it sounds bizarre, but if you shine light on an organism, right, it moves in a particular direction. These algae don't have eyes, but they have phototransductive genes that know like where light is versus where light is not. So when you shine a light on them, that gene becomes activated. Channels on one side of the organism get activated, and it will migrate toward the light. Okay? And that's what you're actually seeing here at the very top. This algae, it's just a petri dish full of algae, and you can see that over time, over 3 minutes, if we shine light over here on the side, that they're migrating over here. If we keep that light on by 6 minutes, you can see a really strong concentration. Most of the algae have moved over here. Okay, so again, a couple of other things to make note of, and you can actually see Carl Dijsselroth, he's holding these different optrodes. We have different wavelengths of light that we can use to stimulate these different channel rhodopsins. We have blue, yellow, orange, etc. Just to make sure that you are aware, the channel rhodopsins that I've just mentioned to you are the blue wavelength open up the channel rhodopsins, the CJH332s. They are generally excitatory. The longer wavelengths, and you do not have to memorize any of these wavelengths, like 470 or the 580, 590, you don't have to memorize these. The longer wavelengths, like the yellow and orange, if a neuron is expressing this receptor, the NPHR, the halo rhodopsin, the HR stands for halo rhodopsin, then they will actually cause inhibition, usually by the influx of things like chloride, etc. And again, this shows this in more detail, that the channel rhodopsins are a non-specific cation channel, influx of sodium, influx of calcium, efflux of potassium. You don't need to worry about any of the details down here on the bottom. And then we have inhibition through molecules like NPHR or the NP halorhodopsin or the archaeorhodopsins, which you see here on the right. And these act to inhibit whenever light is shone on them. This will allow chloride to flow in, causing inhibition, or when orange or yellow light is shone onto archaeorhodopsin, it will cause the efflux of potassium. It is more like a proton pump, in other words, allowing proton to leave, and as a result, causing more negative charge to be left behind. So you can actually do different combinations. You can express both channel rhodopsins and halorhodopsins in the same neurons. And one of the things that you should make note of, if this is a neuron at rest, and this neuron has somehow, and we haven't talked about this yet, started to express these channel rhodopsins, normally it has no effect on them. So this neuron can fire an action potential, then goes back to baseline at minus 70, the resting membrane potential. It can then fire another action potential, again, not something that we're controlling. But if we were to take this neuron and shine blue light on it, we would now see that whenever we shone blue light, shown by this little dash of blue at the top, that now we can actually influence how this neuron is going to fire an action potential. It is no longer going to be random. It is going to follow the pattern that whenever I turn on a blue light, it will fire an action potential by allowing these different ions to flow through. I can also cause inhibition by turning on orange light if indeed we have the expression of channels like NPHR or other halorhodopsins, that whenever I have an orange light on, even if I have excitation going on, that this orange light will completely inhibit, therefore no action potentials during the time of this orange light activation of this particular neuron. Do you have any questions on this general concept or general idea before I go on? So blue light, excitatory, channel rhodopsin, orange light, inhibitory, halorhodopsins or archaeorhodopsins. Yes, Harrison?
Does optogenetics change how long the neuron stays in the refractory period?
So that's a great question. Does it affect the absolute refractory period of the neuron? It does not, but that's a great question. Katya, do you have a question?
Yeah, just when you have the channel rhodopsins and the halorhodopsins, they're just interspersed like equally across the axis.
Yes, so this is a very diagrammatic, cartoon-like figure over here on the right, and it looks like they're segregated in different compartments, but they would be spread out equally over the entire surface of the neuron. It wouldn't look like half of your neuron has channel rhodopsins and half of them have like halorhodopsins. Yes, another question?
Yeah, and then I was wondering, if you shine blue
which is what is happening here. Yes.
Well, I was wondering then, are the channel rhodopsins still have sodium and calcium coming in and potassium going out, but it's just not enough to counteract what's happening?
So that's a great question as well. So what's happening when you have both blue and orange light? So you are activating both channels, channel rhodopsin and halorhodopsins, but the halorhodopsins dominate. Okay, and it's because of the kinetics of the halorhodopsins. You can see that we have to actually stimulate for a longer period of time. They're not quite as good as the channel rhodopsins, even the newer variants, which are on this particular slide. I do not need you to memorize any of this down here on the bottom, nor do I need you to really spend a lot of time on what's up here at the top. Really, it's just to reiterate that we have channel rhodopsins and halorhodopsins that are involved in excitation or inhibition, that we have specific wavelengths of light that will stimulate channel rhodopsins, which is blue, or we have longer wavelength yellow or orange light that stimulate the halorhodopsins, which are inhibitory. I'm also going to mention in passing, because some of you might take other courses where you will hear about these, that in fact the newer variations of these different channel rhodopsins that have been developed since 2009 are actually also G protein coupled as well. They're not just allowing ions to flow through, they can also make connections with other types of G proteins, other signaling molecules, etc. And some of you will hear, even in third year, about these other opto-XRs that have been developed that work on a longer time scale. Again, the details not being so important. So we've looked at these different channel rhodopsins. And one thing to really make sure that you are 100% confident of is that these channel rhodopsins, when we stimulate them appropriately, the activity depolarizes the cell, it increases excitability. Generally, it causes excitation or action potential firing. I'm not going to take time to go through this movie. If you want, the full movie is available for you on this link here at the bottom on YouTube. This was developed by Nature Videos in collaboration with Dr. Diceroth. So I do need you to know some of the details here. Some of you get very, very nervous about what molecular biology actually means. And in some ways, it's just like math. You just have to know what some of these different symbols mean, and it's no more complicated than that. We've already talked about some of these different proteins here. You don't really have to worry about opto-XR down here at the bottom. This is the longer bistable version of some of the channel rhodopsins. I did talk to you a little bit about these halorhodopsins like the NPHR, and I've talked to you about these channel rhodopsins. These are not genes that are normally found in nature. These are genes that have been engineered by those scientists at Stanford or in other places. But in order for these different genes to form these different types of channels, if it gets expressed in a cell, we actually have to do some more engineering on top of this. In order for us to get this in specific groups of cells, we have to identify a promoter. We have to drive the expression using a promoter for this particular gene, the opsin gene. It could be a channel rhodopsin gene. It could be a halorhodopsin gene. But the opsin part means that it's just stimulated by light. This by itself, this DNA, this chunk of DNA, actually can never enter into a cell easily. We have to be able to deliver this into a neuron or into a cell. And that means that if we make this piece of DNA, which is what we're going to be talking about, we have to actually package it and put it into a virus, because a virus can deliver this gene, but the gene, if it was just like this by itself, would never be able to enter into a cell. We would never be able to form the appropriate protein as a result. So we need to have a packaging material as a virus to be able to package this particular DNA or this gene. There's also a lot of other things that you need to be good at. You have to be good at molecular biology. You have to be good at genetics. You also have to be good at neuroscience. You have to be good at surgery. You have to be able to do some surgery to deliver this to a particular area of the brain and to be able to implant this optrode at the top of the mouse. Now, again, you don't have to know any of this. This is normally how we do these procedures. I can tell you of all the different techniques that I learned while I was a graduate student, the thing that I remember the most was actually my very first animal surgery. And my very first animal surgery happened to be the stereotactic surgery, which is what you are seeing here. You don't have to know all of the details, but you do have to know that we are hitting a precise small target within the brain. We can't actually see the brain. You have to make your best guess that I'm going to show you a little bit later on. But these are all of the different steps that you would have to do in order for you to do this optogenetic experiment. You don't have to know any of this, but it's just to give you an idea of how technically complicated it is to start from the beginning, work through, and be able to deliver a very, very small quantity of virus. 2 microliters is less than a normal sized droplet into the brain. So this precision targeting that I'm going to start talking to you about and going in some more detail is really important. Now I want you to get this overall idea over the next few slides, and I'm not going to ask you about the next few slides in the same detail that I am here. I'm going to point things out to you. So for example, we have a lot of different neurons in the brain. Even in the rodent brain, we have hundreds of millions of different neurons. We want a specific subtype or a specific group of neurons. So out of all of the different neurons, we may only want to have interneurons within the brain. Within the entire interneuron population, maybe we only want the interneurons that express a protein that is known as calbindin. So we have interneurons that will express protein A, GAD65 or 67. We might have another neuron or interneuron that expresses calbindin. We might have a third class of interneurons which might express a totally different protein. So out of all those interneurons, we actually have subtypes of interneurons. If we want to target it even further, we might use only calbindin positive interneurons. And of course, there are different brain regions in which we find this. This is the role of the stereotactic surgery. We have to know what area of the brain we want to deliver them. So for example, we might want to only deliver a gene to calbindin positive interneurons that are found within the cerebellum. And we need to be able to manipulate that population of neurons with light. And can we do this? We do this incredibly efficiently. Now in 2017, this is actually a standard technique in most labs. And you can see this is the cerebellum here at the bottom. It's been marked, the genetic construct which has been delivered to the cerebellum here in the bottom is actually marked with a fluorescent protein, which is why you see the cerebellum lighting up. If we were to look at it in closer detail or with a higher magnification over in panel C, we would actually be able to see that not every neuron is lighting up with the fluorescent protein, and therefore not every neuron has had the gene delivered to it. Only some of the neurons have had this. These are calbindin positive interneurons that are found within the cerebellum. So imagine the level of specificity. How did we target all of these? How did we deliver all of this? How do we make sure that out of all of these other neurons that you can't see in the background, why is it only this population that we are able to deliver this gene to? And this is sort of the topic of the next few minutes of this lecture. Now, before I begin, and this is a really important slide, some of you have had a lot of molecular biology, and some of you, it's been a few years since you've had this. Just like channel rhodopsin and halorhodopsin don't exist in mammalian systems, neither does this system here. This is known as the Cre-Lox system, and this is what allows us to specifically deliver genes to specific groups of neurons. You need to understand this Cre-Lox system, and this is a very powerful method that is able to deliver genes to specific groups of cells or to specific groups of tissues. So the enzyme which is known as Cre-recombinase is normally not found in mammals. We don't find it in rats, mice, humans. It's normally a bacterial system. And the LOXP elements, which are part of the DNA, these LOXP elements are normally not found in mammalian systems either. We don't have any LOXP in our DNA. So even if I were to have Cre-recombinase somehow floating around within my neurons, nothing will happen to my neurons because the LOXP there is nothing for the Cre recombinase to bind to. My DNA, your DNA, the mouse DNA, the rat DNA, none of the mammalian systems have these LOXP sites. So what are these LOXP sites? These LOXP sites are simply stretches of DNA that are illustrated for you here. And these LOXP DNA sequences are 34 base pairs long. And these are the sequences that will bind to the enzyme known as Cre recombinase. We don't have Cre recombinase, therefore you have to think a little bit about where is the Cre recombinase coming from. And these LOXP sites are where the Cre recombinase will be binding. I'm going to tell you where the Cre recombinase comes from later on, but is that concept okay for you? These LOXP sites are where Cre recombinase binds. That part's good? All right. Because this is DNA, and you know that DNA has directionality. Normally we talk about DNA being 5' to 3'. So 5' here on the left, 3' here on the right. This LOXP site, the 5' end here and the 3' end is over here. The same thing for this LOXP site, 5' here, 3' on this end. But we can actually write DNA however we want to. I could actually rewrite your entire genetic sequence backwards. I could take your DNA sequence, could actually invert the DNA sequence and make a mirror copy of you simply by reversing the order. The 5' now becomes the 3'. And these LOXP sites can also be engineered in exactly the same way. So we don't always have to have LOXP sites in the same orientation like you see here. We can actually cause them to be in the opposite orientation, and then this opposite orientation has a very different feature associated with it. So here's 5' to 3', and now this end has the 3' end and the 5' is over here. These are in the opposite orientation. Is that okay for everyone? So why do we need two different orientations? Because the DNA that is normally found between these LOXP sites can be manipulated differently depending on what we place within them. So for example, if we were to have this piece of DNA and we were to engineer two LOXP sites in this orientation, the same orientation around them, in the presence of Cre recombinase, what happens is this chunk of DNA that's found in between them actually gets removed from the gene, or the gene gets removed from the piece of DNA. If we had these opposite orientation LOXP sites, as you see here, the stretch of DNA that is normally found within them actually gets turned around or gets reversed or becomes a mirror image of what is normally being expressed. Now, I know that might not make a lot of sense to you right now, but I need you to make sure that you understand this before I go on to the next slide. Do you have any questions or things that you're unsure about in this particular case or this particular scenario? Two different orientations of the LOXP. In either orientation, these LOXP sites, we always need two. You need two LOXP sites to bind Cre recombinase, that when the Cre recombinase binds, what happens to the DNA in between them actually is very, very different. That's okay for everyone? Do you have any questions at all? Or things that you're not sure of at this point? Okay, so this is an important slide for you to go back and review. Now I'm going to come back and talk to you about this in a few moments. This was actually one of the first constructs that was created by Carl Dijksterhoff's group. The only reason I am putting this up here right now is that if you were looking at their construct, and I'm going to talk to you about this in a few moments, but these are the LOXP sites that you see. Okay, now you tell me, are these in the same orientation or are they inverted? They are inverted, so they're facing each other, in other words. And if you're taking a look at this particular gene that's found in between them, this is channel rhodopsin, and we have attached an enhanced yellow fluorescent protein. We genetically construct the channel rhodopsin. We also fuse directly to channel rhodopsin this enhanced yellow fluorescent protein, which is another version of GFP. You guys have all heard of GFP, correct? So wherever we have channel rhodopsin, we also have fluorescence. Now, if you're taking a look, this is actually, this sequence that's found in between is actually already reversed. Okay? So it's already written in the inverse orientation. If I were to write something, it would look like this. Complete garbage or nonsense. In the presence of Cre recombinase, though, if we were to somehow get Cre recombinase, one of the things that would happen is that the sequence that's found in between, just as I shown you on that previous slide, this sequence that's found in between becomes flipped over, or it goes in the opposite orientation. And if we were to do this, then the gene would actually make sense. And so this is sort of the strategy that many individuals in molecular biology take, that these Cre recombinase binding sites, the LOXP, actually give us a lot of specificity. So before I go on, can you tell me why might this be useful? Why have this step? Why bother? Like, why not just go and make something that is going to have this channel rhodopsin? That's what we really want, right? We want to deliver channel rhodopsin. But why have this really weird step of having LOXP and then also having it reversed? Yes?
Maybe so that it doesn't do anything until you get to the location where you want it to be.
So as Katya was mentioning, what if I missed my target? What if I was like James Milner and Peter Milner and James Old, and I'd missed my target? I was completely off base. And what if I didn't want that gene being expressed until it hit the appropriate target? By having it inverted, even if it got somehow into the neurons that were off target, not the ones that I wanted to, I missed, and even if it went through the EF1 alpha promoter and the neurons started to express this protein is complete garbage. There's no sequence to it. It means nothing in terms of the overall sequence. It's not until I get it into the right cells that it will produce the channel rhodopsin. So this gives me that added security or that added level of specificity. Now, we haven't talked about this in a lot of detail, but I'm going to also mention to you that, and some of you have dropped by the office to chat about neuroscience in general. Neurons are protected by the blood-brain barrier, but one of the things that actually makes that blood-brain barrier somewhat obsolete is this concept of neurons being neurotropic. Do you know what the word tropic means? I'm not talking trophic with an H, but tropic. Anyone taking that Classics 201, that supposed bird course in arts and science? Tropic? Tropic? Any ideas? So this means that it moves toward, and these viruses are very heavily attracted toward infecting neurons. If you think about the rabies virus, for example, you get bitten out here in your hand. How does that rabies virus here actually affect your brain all the way up here? It's because viruses are very neurotropic. HIV, HSV, any virus that you can think of, they're all very neurotropic. They like infecting neurons, in other words, and so we use that to our advantage. Can we package package genetic material, which is what viruses do anyways, and use the viral part to deliver our payload or our gene to specific groups of neurons? And I'm going to go through this one slide and then we'll take a break in case you have any questions. So if I were to take a look at these different types of viruses, these are the ones that are most commonly used in science, in neuroscience and cell biology. If you're going through this purple sheet, you're going to need to know some of this information, not just to memorize it, but to actually be able to apply it based on what you've learned. So why have different types of viruses? So for example, the adeno-associated virus, which is the most common type used in neuroscience, the genetic material that we use and package into the virus is usually single-stranded DNA. It's easy for us to work with single-stranded DNA. We can do PCR incredibly quickly with single-stranded DNA. However, we're limited. That DNA that we can package into an adeno-associated virus can't be much bigger than about 5 kilobases in length. So what we put in is limited to about 5 kilobases in length. We also know that it takes time. If I were to somehow create this DNA construct and I were to package it inside an adenovirus, I was to inject it into an area of the brain, then I went back and looked for channel rhodopsin, I wouldn't actually see any channel rhodopsin until several weeks had gone by. I have to wait until this virus actually expresses this particular protein. However, once it's in that neuron, that gene that has been delivered through the adenovirus will last years. I'll be able to track it for years at a time if I use an adenovirus. Same idea for lentivirus. My starting material has to be RNA. It's a little bit bigger. I can deliver about 8 kilobases worth of RNA through this lentivirus. It also takes time, and it also will last years. And the herpes simplex virus is actually kind of unique in the sense that it uses double-stranded DNA. I can package a lot into herpes simplex virus. I mean, think about that herpes virus that you read about, those little cold sores that appear. They last in neurons for a very, very long period of time. And these different DNA can actually be quite big, about 150 kilobases. If I want to package more than one gene, this is what I would normally use. However, there's some drawbacks, like you're thinking, why not just use this all the time? So even though the speed of expression is fast, so I infect a group of neurons, I deliver this virus into an area of the brain. I wait a few days, 2 or 3 days after my surgery and after delivery. I'll start to see channel rhodopsin or enhanced yellow fluorescent protein. But generally speaking, this doesn't last very long. So it goes on really quickly, but after a few weeks, maybe a month, the herpes simplex actually starts to disappear. We don't see that lasting for long periods of time. Again, there are things that people are trying to do to increase persistent or long-lasting expression, but it doesn't seem to be as effective right now. So I think this is a good place to take a break. I'll just leave this slide up here just to give you an idea of all the steps. I'll take you through this really quickly. You don't have to worry about this slide. I usually share this with some of my fourth-year students when we talk about actual viral packaging, but it's there for you to know all of the different steps that are actually involved. But we'll take a break and we'll come back and continue on with the rest of this.
What point do you actually put it in the mouse? So how old is this? Like when they're still when they're not.
Depends on whenever you want to do a specific behavior or when you want to test. It can be any.
Because wouldn't it, like for these ones, if they take weeks to actually be expressed, wouldn't it be useful then to put it in the mice before they even born?
Usually we put it into young adults.
Okay, so on the part of molecular biology, if they're facing the same direction, what you do is after pre-recombinase time to chop off the entire target, right? Which means that
Well, you're still left with one
so
molecule.
one gets chopped off the DNA. For inversion, what happens if they're initially the lock speed, if they're inversed, so initially this is 5 to 3 prime, right? So you just inverse it 3 prime to 5 prime 3 prime the other direction. So you basically flip the direction. And as a result, we use this technique because you have to first
Go through that, Andy, but it's basically in case you're off target, that gene that gets expressed is actually not going to make any sense. It's not going to affect the cells. You're not going to see any physiological or biophysiological
So
consequences.
it's just there as an insurance because make sure even if it's even right here, because there's no lock speed, it's still in the other direction. So it's just jump, right?
Yep, still just jump.
You need to know what you're sign off for?
And I talked about all of that.
I had a question on that as well. Doesn't that only actually help if you assume that you express Cree in the right spot?
I haven't talked to you about Cree yet, but
Okay.
this is the Cree driver animals that I mentioned at the very beginning.
Okay.
That's a good point. But
Yeah.
give me some time, Armin. I'm getting there.
Alright, yeah, I was
Getting
just
there.
like,
Don't
yeah,
don't
okay.
jump the gun quite
Okay.
yet. But good points. Yes.
I have a quick question about these so I'm not quite able to connect how you use the virus to differentiate between different areas.
It's not used to differentiate between a recent friend. You have to design all of this ahead of time. So for example, some of these questions I want to deliver to a certain area. So I'm gonna go through like the stereotyping Oh, okay. Yeah, because I was only confused. Every time that you target the place, the neurons that you want to do in terms of where you want to plant the virus could affect, the virus theoretically could go somewhere else or bind to another neuron that you may not. But when I go through all of that for you, sorry, that's really common. Sorry? Common. Oh, that's good questions. Just give me a few minutes and I'll take you through that.
Okay, also, just one other thing, if you can, please drop by to go over your term test too. I'm happy to take a look at them. You should probably try to do so after lecture today if you're still on campus. I know it's not the posted hours, but I had made a mistake on that. So immediately after lecture, if you want to head back, you're more than welcome to go over term test 2. The same thing for Wednesday's class. There will also be some times posted for Thursday, but I do want everyone in class to actually go through their term test, make sure that your final exam, like you know what the expectations are, and if you can do that in the next week or so, that would probably work best. So let me just take you through this one slide that you don't need to know in any detail really quickly. What we're trying to do in the next little while is to actually develop this plasmid. When we talk about a virus, and it's easy to just conceptualize, oh, you make this piece of DNA and then you put it into a virus. That's not quite exactly correct. What you have to do is, you know, for a virus actually isn't a living organism by itself. So we have all of these different viral constructs that will form the envelope and will form some of the different binding proteins. Then your DNA of interest that you are creating yourself. Channel rhodopsin goes here. You have to take all 3 of these, then infect HEK cells, and these HEK cells, after they get infected, will then form the virus. And again, you don't need to worry about any of these different details. Once you form the virus, you actually have to purify the virus because it kills these cells. You are left with the remnants of these dead cells. You need to purify the virus and separate them from the dead cells on the bottom. You do this by ultracentrifugation, either on a sucrose gradient, or you can use a cesium chloride gradient for different types of viruses because they're different sizes. And then ultimately, you're left with this small amount of stock that you actually have to then aliquot out. That's the process. It's not really all that simple. It's a prolonged process. Many times this doesn't actually work. You might go through this entire process, create this construct that you have up here, which we're going to be working on next, and then at the end of it, when you've put it into the brain of a mouse or a rat, it doesn't work. And you have to go back to the starting blocks to figure out why. And sometimes when you're doing this stereotactic surgery, to do all these experiments, you have to actually be really good at a number of different techniques. If you ever have the opportunity to join a lab that will allow you to do all of these, molecular biology, animal studies, animal surgery, electrophysiology, all of these things work together. There are very few labs here at U of T that actually have the capacity to do all of the things that I'm mentioning to you. So I mentioned at the beginning that we have to deliver this to specific groups of neurons. And it's really hard because we don't get MRIs of rats, we don't get MRIs of mice. How do you know where you're actually delivering your virus to? This gets back into stereotactic surgery. And I do need you to know some parts of this. I don't need you to memorize all of this. So one of the things that we have is either for a mouse or for a rat, we have a roadmap. It's called an atlas. And this atlas is based on a number of sections, which is what you see here. And these sections that are shown on each of the pages of the mouse brain or rat brain atlas are stained for Nissl or Kressel violet, which is why you see this. We are able at every section to identify key structures. So just based on this particular staining here, for example, you'd be able to identify the beginnings of the hippocampal formation, etc. But again, that's the brain, and that's the brain part. But we actually don't see that. If you go back several slides, what we only have is sort of this, the top part of the rat. We don't see any of that in this particular picture. So how do we know, based on this map, where we're actually at in the brain? So if you were to take an animal and put it to sleep, and then you were to remove the fur, and then you were to cut back some of the muscle on the top of its head, you'd actually be able to get down to the level of the skull. And on the skull, just like you have a number of suture lines on your skull, and the rat and mice also have suture lines on their skull as well. And again, I'd like you to make sure that you know these major landmarks. So for example, near the eyes, and these are the eye sockets, we would have a structure known as bregma. And this is that suture line that you see here. The different coordinates, just like GPS coordinates or Google Map coordinates, are based on either bregma or they're based on things like lambda, which is the other suture toward the back of the brain. So this is the other suture line known as lambda. And so if you were to take a look, if I wanted to know, using stereotactic surgery, I wanted to hit a target known as the VTA, or the ventral tegmental area, I would look this up in an atlas, like the mouse brain if I was using mice, or the rat brain, and it would actually tell me where to drill my hole. So it would tell me, for example, that I should go from bregma, this line here, I should move back 3.4 millimeters backwards from bregma, which is this number here, and then laterally from this suture line that you see here, laterally toward the side, I should be moving 0.35 millimeters, and then how deep should I go? I should be going 4 millimeters in depth from the surface of the skull. No? Yes? Okay, cool. All right, awesome. So this tells you with precision, without doing an MRI or anything else of the brain, and these numbers aren't ones that I need you to memorize, but I want you to be familiar with how this is done. That we would drill a hole in this area, and we would then put in our electrode that's going to deliver the virus to these precise coordinates. If we do so, and we are being precise, then we should be able to hit on every single experiment the VTA at exactly the same coordinates. And that's just the stereotactic part of it. Now I'm going to ask, do you have any questions before I go on, or anything that you're curious about? Have any of you done stereotactic surgery? No? It's a very difficult procedure. I was really lucky when I started my master's project. I actually got involved in doing stereotactic surgery first. I was taught by an orthopedic surgeon, so I was taught like proper surgical techniques. And I remember the proudest moment I ever had, because I always wanted to impress my postdoc, was when I had this animal set up in the stereotactic rig. It looked perfect to me, so there was no bleeding, nothing else was going wrong with it. Every other experiment I had, there was like tons of bleeding. This was like a perfect stereotactic surgery. So I ran over to get my postdoc. I said, come over here, you got to take a look at my beautiful experiment going on. And he takes a look, and he says, this is beautiful, Bill. And I said, thank you. I'm really, really proud of it. And he goes, and you notice there's no bleeding, right? And I go, yeah, I know. Isn't that amazing? Like, I'm better than you are. And he goes, yeah, do you know why the animal is not bleeding? I said, because I'm good. And he goes, no, the animal has died. And I remember feeling so embarrassed, because I hadn't even thought that I might have killed the animal inadvertently during that procedure, which also does happen as well. So that's sort of the big picture. Now I need you to start thinking about this. So we're going back now. You're going to create this. Now the stereotactic part, where to deliver it, is clear. The fact that we need to create a virus and that different viruses have different sizes, hopefully is something that you'll spend some time going over and memorizing, that HSV is bigger than AAV and different than lentiviruses, etc. But I need you to start thinking about that construct that looks a lot like this. So again, when we're talking about DNA, there's always directionality. By convention, this is 5' on the left and 3' on the right. So you guys all know that these are LOX-P sites, and these LOX-P sites are non-symmetrical here, and that in the presence of Cre recombinase, whatever DNA sequence is found between it will be inverted or flipped over. That part hopefully is somewhat clear to you now. But how do I make sure that I am delivering my construct, the thing that I'm working on? How do I make sure that it's only going to be expressed in dopaminergic cells? So this gets to the very top of this pink sheet. What promoters can I use to deliver and make sure that it's only being expressed in cells that are dopaminergic? Well, again, this relies on the fact that you know a little bit about neurobiology. So it might mean that you have to use the promoter for tyrosine hydroxylase, which is only found in dopaminergic cells. Or if I want to only deliver this to either excitatory or more specifically glutamatergic cells that are excitatory, that I would have to use the promoter for something that's specific for excitatory glutamatergic cells, like the promoter for glutaminase. That means that the promoter, this thing here, will deliver this gene here only to glutamatergic neurons. So I could use glutaminase promoters, or I could use VGlut2 promoters. Whatever it is, it will be specific to those glutamatergic neurons. I may decide that I want to drive this in inhibitory or GABAergic neurons, in which case I might use things like glutamic acid decarboxylase promoters to make sure that only neurons that express glutamic acid decarboxylase will also express the gene that I want to be able to deliver. Is that okay for everyone? So I could use any of these specific promoters like TH, glutaminase, or VGlut promoters, GAD, or I could use VGAT, which is a transporter that allows GABA to be put back into vesicles. I could use any of those. Or oftentimes what happens is we use a general promoter. So it doesn't matter what cell it goes into. I want it in like every single cell. I don't really care. In this particular case, we might have things like the CMV promoter. And this CMV promoter, which is a cytomegalovirus enhancer B-actin promoter, this CMV site will express it in every cell. It'll do neurons, it'll do glia, it'll do like any cell that will take up DNA. Or we might use elongation factor 1 alpha, which is what we started off with over here. The EF1 alpha is just a general, non-specific, any cell, if it takes up the DNA or gets infected, any cell will express whatever comes after that promoter. So the promoter only tells the cell to make whatever comes up after it, and the promoter allows it to be in specific groups of cells. Do you have any questions on that? Because it's a really important concept. This allows us to target specific cell types. This is that very first question, or the scenario 1 questions that you see there. You should be able to identify how you're driving it in different cell types. This is, as this says here, very important. So make sure that you are aware of it. The key question is, though, that I was asked by Armin and others during the break, how do you get this Cre recombinase? How does that work? We haven't talked about Cre recombinase. I get this LOXP. I sort of understand this promoter, and the promoter always comes first. By the way, when you're asked to draw this out, and it will be one of the questions that you will have, you always draw the promoter first. And this is one of the things that I want to make sure that no one gets wrong this semester. Promoter always goes first. Then you generate the LOXP sites usually after it. And then you put the gene of interest in between, and then you close it with this other LOXP. Don't put LOXP in front of the promoter. Don't put it like anywhere else. Promoter always goes first. So how do we do this? How do we actually get this Cre recombinase? I haven't told you about that. I've only told you LOXP doesn't exist in mammalian systems. I've also told you Cre recombinase normally doesn't exist in mammalian systems either. And one of the things that I've neglected to tell you is that we have what are known as Cre driver lines. We have mice that have been created by different transgenic facilities that already drive the expression of Cre recombinase in certain groups of neurons. So, for example, we have what is known as a TH, internal ribosome entry site, or IRES Cre recombinase, which means that Cre recombinase is only expressed in neurons that express tyrosine hydroxylase. What is that specific for? Tyrosine hydroxylase is specific for dopaminergic neurons. So Cre recombinase, in other words, is specific for dopaminergic neurons. That mouse already exists. That is a Cre driver line. I could have a Cre driver line specific for VGlut2. Where would I expect to see Cre recombinase being expressed? If I have a VGlut2 IRES Cre recombinase. Glutamatergic types of cells. Okay, perfect. So again, this is just a proof of concept that if we have the VTA and the substantia nigra, both of these are dopaminergic, and you were to look at a wild-type mouse and you were to stain for Cre recombinase, it normally doesn't exist, and therefore we get no green staining here whatsoever. exactly as we would expect. If we were to then take a look at the VTA, and then we were to take a look at the substantia nigra as well, and we were to take a look at the staining for Cre recombinase, now we actually see green staining because this mouse actually has Cre being expressed only in the dopaminergic neurons. As you see here, we get the combination of the two. And this is one of the ways in which we get this specificity that I showed you earlier. How do we make sure that only specific neurons express these different channel rhodopsins or these different GFP variants, etc., by using these different Cre recombinase driver lines? Any questions on this before we go on? Okay, and again, not to spend too much time on this, we can do a number of different things. This isn't, by the way, you should not worry about this particular paper, this particular figure. You don't need to memorize it. It's just to give you an idea of the ways in which we can use this specificity. So we can deliver, for example, with high levels of precision now, even though these neurons are really close together, we can deliver our gene of interest using the systems that we just talked to this neuron, but not this cell, and to this neuron, but not this neuron over here. This gives us a lot of specificity, even in this really small area. We can also allow the channel rhodopsins to express down the entire length of the axon, not just the cell body, but also within the dendrites and axons, etc., which means that we can also stimulate not just the cell bodies, but also the axons directly as well, with the same level of specificity. We can go back and test different types of behaviors using this, etc., which is why everyone's so interested in this optogenetics. Now, I'd shown you this particular paper or this particular figure earlier. I just wanted to also highlight this particular aspect, that if you are looking over here on the right, a stain that we normally use, and I've asked that it be included in every single HMB third-year lab because it's a really easy technique. It's just like when you were staining your DNA gels in first-year biology that you used something like ethidium bromide or something similar. Did you do that experiment? Did you guys stain your DNA gels? Do you remember what you used, like cyber green or something else like that? Don't remember? It's okay, not important. This DAPI, which is in blue, actually stains all of the cell nuclei because it's specific stain for DNA. Doesn't matter what type of cell, all cells contain DNA. It could be glial cells, could be neurons, whatever cell it is, as long as it has DNA, it will be stained by DAPI, which is a simple histochemical stain. And that's really all I want you to take away from this slide, because I think it's important when you're leaving this course to have some idea of what DAPI is all about. Okay, so let me take you through this step by step. Why talk about optogenetics? So I need you to realize a couple of things, and just to orient you on these next few slides. These cells here are Th positive, therefore what type of neurotransmitter do they have? These are dopamine. These cells also specifically, because it's a Cre driver line, will express Cre recombinase. Is that clear? So only the ones here in light blue have Cre recombinase. It's good for everyone? Let me ask you something to make sure that you really understand. Is that going to affect this group of cells, the ones in light blue? These cells have Cre recombinase. We normally don't find Cre recombinase. Cre recombinase does a whole bunch of weird things. Does it normally affect these neurons, these Th positive neurons? Max, you're shaking your head no. Why?
We haven't infected them with the virus.
Okay, so no virus. But okay, what does Cre recombinase do, just generally speaking? It can excise DNA, it can flip DNA. Will it do that to the DNA in these neurons? It will not, because normally, even though we have Cre recombinase all over these blue neurons, and only in these blue neurons, because it's being driven by the Th promoter to make Cre recombinase specific, that this DNA does not contain LOXP. There is no naturally occurring LOXP in the mammalian nervous system. So even though we have Cre recombinase, it does nothing to these cells, okay? We also have these other cell types all around it, but we want precision. We only want to be affecting this because maybe we want to study this group of cells and how it interacts with this target, like the nucleus accumbens. Is that okay for everyone as an overview? Cre recombinase, it's there, we don't care. I like that. That's a good rhyme. All right, so now let me take 2 microliters of this virus. I found the right stereotactic coordinates. I've sat there, I've kept the animal alive this time. I've drilled through, I've put the microinjector right above this area, and then I very, very, very slowly infuse this whole area with the virus that I've created. And this whole area gets flooded. This entire area gets flooded with this virus that I've created. Every cell here is going to be exposed to this virus, okay? So if I were to do that, and then I were to tell you a little bit about the virus, so let me take you through a couple of things that I want you to understand, because I don't want you to simply look at these different figures. I want you to understand them whenever you see them in your 3rd year cell molecular courses or in neurobiology courses, etc. If I am generating something that goes into a virus, I actually have to help the virus a little bit. And so for any viral vector, anything that gets packaged into any kind of virus, you're going to see these little things known as internal tandem repeats. These ITRs sit at the end of your construct. I am making this, so I'm actually planning ahead of time to have these internal tandem repeats. If I see that, that means that this DNA is going into a virus. I don't need it, but if it's going into a virus, I need these ITRs. And you'll see that on every paper that you read. You'll never be able to read a paper again the same way. Anytime you see a TR, whether it's LTR or ITR, you will know that it's going into a virus. Then I'm going to use this particular promoter. I'm going to use the EF, or elongation factor 1 alpha promoter. What do you remember about that promoter? Is it specific to dopaminergic neurons? No, it's just a general promoter. All cells, when exposed to this promoter, will express the gene that follows after this promoter. So now I'm creating some LOX-P sites. I am engineering this myself. So tell me about this LOX-P site. Is it symmetrical or is it non-symmetrical? What do you know about it? So these are inverted, which means that the sequence that's normally found in between is going to be inverted if it encounters Cre recombinase. So this is the type of thing that you need to be able to tell me about on your final exam. Again, I've been doing this for a number of years, so for me it's fairly straightforward, but I know that some of you, it may be fairly new or you haven't encountered it in a while. So there's no need to tell me right away, but do you have any questions right now? Or if you think about it later, come and see me, but anything that you're curious about right now? Yes, Luke.
So ITR is the virus-specific promoter.
Correct. So different viruses will take different types of tandem repeats. So you'll have an LTR or an ITR, but if you have this on either end of your construct, that means that it's going into a virus. Okay, and you'll see that, and I'll point that out as we go along. Anything else? Okay. Yes, Summer. So right now, we only have one, and it's the promoter that we're going to use to infect, and we just have infected this whole group of neurons that we see here. Previously, we've had a promoter in a separate scenario that will drive the expression of Cre recombinase only in Th-positive cells, which is why they're in light blue, okay? But yes, originally when they designed it, they would have used a separate promoter to make sure that it was only that group of neurons that express Cre recombinase. Any other questions? Anything else at all? Like anything you're curious about, not sure about, wondering about? Yes?
The ITR, is that like classified?
These are just short stretches of DNA that actually help the virus to be incorporated into genomic DNA. So these are tandem repeats. Okay? Again, those details not so important. The ITR being found in a viral vector, yes, but how it actually works, not so much. Any other questions? Okay. So I have packaged this virus into a virus, which has then been delivered to all of these different cells here. Okay? So this is the region in which the virus has been spread around. Isn't this like exactly what I started off with by telling you that, you know what, when we put in an electrode and we stimulate electrically, it creates this little bubble. All the cells within that bubble get stimulated. Why is this any different? I'm going to try to convince you that this is indeed a little different than what I mentioned to you earlier. So let's think a little bit about this. So if I were delivering this construct in the box to all of the neurons within this sphere that you see here, which of these different neurons will, or these groups of cells, I'm not specifying neurons per se, but which of these groups of cells and or neurons would actually express this promoter? Where would I be able to detect this promoter? Which of these cells? How many of you are saying all of them? All of them? How many of you are saying just these? How many of you aren't saying? Nice. One honest person. Awesome. So actually, this promoter gets expressed everywhere. All cells will express this gene. Every last one of them gets infected. It's promiscuous. This EF1 alpha promoter just goes into every cell. It will try to make this, but of course this is a nonsense sequence, right? It doesn't matter if it gets in there. The promoter will drive the expression of this, but this is upside down, inverted. It doesn't make any sense. It won't code for a functional protein or a functional channel. But let's think a little bit about what we've actually done. So these cells, which have also been infected, contain Cre recombinase. What will happen in the presence of Cre recombinase to this particular gene? The way that these LOXP sites are, what will happen? It will invert it. So it will put it into its correct reading frame. So again, we normally put things like EYFP into these cells to make sure that the gene is being delivered. We want to be able to see it, just like the fluorescent cerebellum, etc. So if we were to see that this is a fluorescent protein, which of these cells would actually fluoresce green? Would all of them fluoresce green? Which cells would fluoresce green, and can you tell me why? The ones that express Cre. Okay, so these would be the ones that we would expect to see having the EYFP, which is shown here in green, and it would only be this. So even though we have infected all of these different cells and had this P promoter, the EF1 alpha, being infecting all of these different cells, only in these cells, because these are specifically expressing the Cre recombinase, only these cells are the ones that we will actually get the correct orientation of the gene and therefore get the correct expression of the enhanced yellow fluorescent protein. This was a really important concept. I know some of you look like you are thinking it through. That's okay. Thinking it through is fine. Do you have any questions over the next few days? Because this will make up a big part of your final exam. Being able to answer all of the questions on this pink, purple sheet will be important. Please make sure that you're doing this as early as you can. It's really not that challenging if you're spending time on it, but if you're leaving it to the weekend before your final exam, it is going to be somewhat challenging. So we mentioned in a previous slide that we don't just do this for electrically stimulating or optogenetically stimulating. We can also look at behaviors. And this is actually Carl Dijsselroth's first paper. So it was actually submitted in 2008. It got published actually fairly quickly over the course of a few months. You don't need to know this particular paper or anything from it, but this is the very first paper that was published in Science that actually looked at behavioral conditioning, and it actually used optogenetics. It was actually one of the most cited papers that are out there in terms of optogenetics. I've already shown you this. This is their construct that they used in this particular paper. So if you're taking a look at this, and again, you don't need to worry about all of the details, you already know what this is. You know that this is an EF1 alpha promoter. You know that these are LOXP sites. You don't have to worry about the LOX 2722 sites. Those are different. If you take another course in cell molecular biology, you'll learn a little bit more about this. But we don't have to worry about it here. And you can see what the orientation is like. You'll also see that we have these ITR sites. In case some of you are wondering what these WPR repeats are hereafter, once you've created the protein, to stabilize it, we often add different elements like a poly-A tail. We'll add things like the woodchuck hepatitis virus transcriptional regulatory element, or the WPRE. And all of this helps with the RNA becoming better transcribed. Don't need to worry about that. I'm not going to ask you about it in case you were really curious. But I do want you to make sure that you understand how this works and where this Cre recombinase is coming from, given the fact that we have a TH, internal ribosome entry site, Cre recombinase mouse line that exists already. So we are using that mouse now to allow for this Cre recombinase to drive this expression, which will put it into its correct orientation. Now, I'm not going to spend a lot of time on this, and it suffices to say that this is the first figure from the Diceroth paper, and what they do is they do exactly the same things that I've outlined for you. They use stereotactic surgery. They isolate the coordinates using Bregma and using the coordinates from the atlas. They go down 4 millimeters. They deliver the virus here to the VTA, and they are using these TH Cre recombinase mice. So these mice have Cre recombinase everywhere where there is TH. The VTA has dopamine, therefore it has TH. Do you have any questions on that part before I go on? So one of the really important things is that the VTA will send its projections. These are the axons. The axons go to a number of different structures. The VTA neurons will send axons up to the habenula. It will send axons to the striatum. It will send axons to the nucleus accumbens. It will send some to the amygdala, some up to the prefrontal cortex. It will actually even send projections all the way up to the hippocampus. Based on everything that you've learned in psychology as well as your first or second year neuroscience courses, which of these targets is the most important for addictive type behaviors? The nucleus accumbens. And that's something that should be ingrained in all of you. And this nucleus accumbens is the area that Carl Diceroth actually wants to be able to look at. So again, you don't need to worry about all of these different details. His paper was the very first paper to actually use optogenetics. So he had to prove that it actually works. And so again, not to go through this in too much detail because you don't have to worry about it. But once he saw channel rhodopsin positive cells, this one here, so this has channel rhodopsin, it has enhanced yellow fluorescent protein. If you were to shine blue light on it, he would actually see that there is this big inward current, and it lasts as long as the blue light is shone on it. And again, he can also manipulate these neurons by flashing on blue light whenever he flashes on blue light. So every time he flashes on blue light here, he gets an action potential. And these things look a lot like dopaminergic, triphasic types of activity. Now, why is he doing all of this? And again, you don't need to worry about all of these details. You can't just publish a paper to demonstrate a technique. And he was actually doing all of this because there was a big debate in neuroscience as to whether or not, if I am looking at a behavior, an addictive type behavior between the VTA and the nucleus accumbens, I can either have like continuous firing, so I can have tonic, steady state firing all the time, or do I get phasic firing, where I get little bursts of firing? Which one is it? In order for me to be addicted to a substance, which one of these two different things actually occurs? No one was able to actually demonstrate because remember, electrodes, stimulation, whole groups of neurons, many different areas, targets, etc. So he wanted to actually use this sophisticated, temporally and spatially specific technique to be able to demonstrate that one type of firing caused an addictive-like behavior and another type of firing did not. And he was the only person at the time that was able to demonstrate that the timing of these different things, because he's not directly stimulating with an electrode, he's stimulating with flashes of light to activate different specific groups of neurons. So going back to the initial experiment, so we don't do these types of experiments anymore. We don't do these Skinner box types of experiments very often in neurobiology or neuroscience. Most of you will be more familiar with this type of apparatus. And again, you don't need to worry about the details that are up here. How many of you have heard of what's known as a conditioned place preference? In psychology? Any of you? So in psychology, for example, we don't do the types of experiments that the McGill group did initially, but one of the things that we can do is to inject a mouse with an addictive substance or a substance that we think might be addictive, either cocaine, nicotine, other substances, and we can keep it in a training chamber as is here. And then if it finds that particular substance that it likes, and it makes this association that this box is somewhere where I've gotten a drug that is beneficial or makes me feel good, then do you think it wants to go back to this box? Yep. Okay, so I would agree that if it was a good experience that you'd want to go back to this particular chamber. And so this mouse is then put into this middle chamber here, and it's given free access. Will it go here or will it go here? Normally, it'd want to go into this neutral chamber. Animals like being in the dark, mice like being in the dark, rats like being in the dark, and this neutral chamber is where it might like to normally go. But it's also got this drug-associated chamber. And what Carl Dijkstra wanted to know is, based on these types of early experiments that many people were doing called this conditioned place preference, could he actually show that the firing pattern made the animal go in one box versus another? And again, he looked at either tonic, continual firing, so little flash of light, wait a few seconds, little flash of light, wait a few seconds, little flash of light, wait a few seconds. And he wanted to know, is it tonic firing that makes an animal go to this conditioned box, or is it the phasic firing that actually makes it go into this box? And it turns out that if you were to do phasic firing, just flashes of light, really, really quickly, as the animal is getting injected with this stimulatory drug like cocaine, that just the flashes of light is enough for it to go into the addictive-like box. So he was able to show, and the details not being so important, he was able to show very clearly that the animal, after phasic training, will stay in the conditioned box similar to as it received a particular drug. So we now can do behavior, we can now stimulate neurons, etc., and these are all very important for us as neurobiologists. And again, if you're really curious about how the experiments worked, I'm happy to tell you about it. I don't want to spend a lot of time on it because, again, we can have a more fulsome discussion if you're really interested, but I don't also want to detract and get sidetracked from what I really want to get across, which is the molecular technique to allow us to study these different types of neurons and synapses. So if you followed everything that I've mentioned to you earlier, these optogenetic molecules are really good for fast changes. They're almost like having neurons and channels directly. But we know that many of the neurons actually express G proteins, which work on a much longer time scale. And this other technique, which is known as the DREAD technique, and this DREAD stands for designer receptor, exclusively activated by a designer drug, and you'll understand all of this in a few moments. This will control neuronal function. Same idea, we have to put these designer receptors into specific groups of neurons. So we need Cre locks, we need promoters, we need all of this. But the gene that's being delivered is not a channel rhodopsin, it's one of these different DREADs. Instead of activating it with flashes of blue light or inhibiting through flashes of orange light, we now use different designer drugs that normally we never encounter as well. So this means that we have a synthetic, not a naturally occurring receptor. We call these the human or HMXDX family, and we can incorporate this into a rat or into a mouse. And again, we still have to decide which cells this is going to be delivered to, but we can also control the activity of the neurons that are expressing this using a slightly different method. And again, this is just an overview. Same idea, that we would be able to have this gene that's being delivered, either the HM3 or the HM4, that is going to be packaged into a virus. It gets stereotactically delivered into an area of the brain that we specifically want to interrogate or to observe. And then instead of using flashes of light, we can then use this designer drug, which again has no effect on any other neurons. Only neurons that express either HM4D or HM3D will be activated by this drug known as CNO. This is a clozapine derivative. It normally will not stimulate any of your neurons, but only neurons that have these specially designed types of receptors, HM4D or HM3D. Just like with optogenetics, this would be similar to channel rhodopsin. The HM3D will cause excitation in the presence of CNO, and the HM4D will have inhibition of whatever neuron it's expressed on when it is exposed to CNO over here on the left. We can either inject this CNO into the mouse or rat, or we can have it in its drinking water for prolonged periods of time, and so we can then see what the overall changes are in behavior. You don't need to worry about all of the details on this slide. I'm going to point out the most important ones. So these are all derived originally from the muscarinic receptor. So muscarinic receptors are the G protein version of the acetylcholine receptor. So we haven't really spent a lot of time on it. But these drugs, like CNO, will not work on our normal muscarinic receptors. They can't activate normal muscarinic receptors. They can only activate muscarinic receptors that have been specifically designed with these little X's to respond to CNO. So nothing else. Acetylcholine, by the way, also doesn't work on these. So acetylcholine doesn't work on HM3D nor on HM4D. Only CNO activates them, and CNO does not activate any other muscarinic receptors. It's very specific, in other words. So I've just hit you with a whole bunch of talk. Do you have any questions on that before I go on? Any questions about CNO, HM3D, HM4D, anything related to any of those topics? So again, CNO, it's long form, and you should use the short form wherever possible. The clozapine N oxide is a designer drug, doesn't interact with any of your normal receptors, etc. I don't need you to worry about these different variants in the rat, which are shown in B and C. The only one that I want you to worry about are the human versions, which are the HM3D, which is normally causing excitation, and the HM4D, which normally causes neuronal silencing or inhibition. Okay, those are the only forms that I want you to know, and I also want you to know what receptor they're originally designed from, the muscarinic, as well as the specific ligand, the CNO, that activates these, but no other types of neurons. Any questions on that? Any questions at all? Okay, so I'm going to leave off with this final slide. And again, the details on here are not that important. I'm going to point out things that I want you to take away from this. Some of this should be somewhat familiar to you now. I'm hoping some of it's somewhat familiar to you now. That when we're looking at different types of viruses to deliver stereotactically using a map to specific areas of the brain, sometimes, and this part you don't have to know, we actually have to know how wide the radius of injection is going to be. Lentiviruses stay within a very small area. Adenoviruses tend to spread out a little bit more, so the spread is important, not so much for what you need to know. But this promoter specificity is something I need you to know. Right now, you couldn't have answered all of the questions that you had on part 1 at the very top. So we haven't really talked about all of these specific promoters. So we always talk about a promoter, small p, and then we talk about the gene that the promoter is working, or the cell type that the promoter is working on. So the promoter for GFAP is only found within astroglial cells or within astrocytes. So this would be how I would deliver a gene specifically to astrocytes or to astroglial cells. If I'm looking at delivery to any type of neuron, I don't care what type of neuron it is, but I don't want it to go to every single cell. I don't want it to go to astrocytes. I don't want it to go to oligodendrocytes, but I don't really care what neuron it gets into as long as it's in neurons. Then I want it to be delivered using this synapsin promoter. And again, this is for all neurons. It's not specific to any type. It's for all neurons. I would use the synaptic promoter. Is that clear for everyone? Yes? So if you were to take a look at this, for example, so a couple of things. So you see LTR, long terminal repeats, over here. Then you know it's going to be packaged into a virus, and indeed you can see that it is going to be packaged into a virus for stereotactic delivery. What is its promoter? Synapsin. So that's going to be expressed in what cell type? All neurons will express whatever follows after it. And here's another thing that I need you to think about. We have a number of fluorescent proteins. Many of you will know of a fluorescent protein like green fluorescent protein. Do you remember the one that we talked about in class so far? So DAPI is a histochemical stain for all DNA, but a gene that we can deliver, what would it be? Well, that's this one. How about a different one? How about something that fluoresces yellow-green? Yep, so I think I've heard it. EYFP, or enhanced yellow fluorescent protein, which looks very much like GFP. And so these are both kind of greenish color. And then we can have this other one that is shown here, which is known as M. cherry. And as the name suggests, it makes whatever cell takes it up to fluoresce red or cherry color if the gene is being properly expressed. So now I hope that you can understand a little bit of why it's in this reverse orientation and why we have these LOXP sites the way that they are. So in the presence of Cre recombinase, this will then cause this to be inverted, which is what will happen. And one other thing, we need to know where that Cre recombinase is coming from. In this particular case, we have a Nestin Cre mouse. Now you have never heard of Nestin, but Nestin is something that we are going to talk about when we talk about stroke. And Nestin is a marker or a protein that is specific to neuroprogenitor cells. So tell me a little bit, therefore, if this is a Nestin IRES Cre recombinase mouse, where is this gene going to be appropriately expressed? Not the anatomical region, but what group of cells? Will I find it in GFAP positive cells? Will I find this M. cherry in GFAP positive cells? Yes? It might be all neurons, but neurons that are progenitor types. Any Nestin positive cells will have the Cre recombinase. So remember, Nestin is a neuroprogenitor cell-specific protein, and only the Nestin cells will have Cre recombinase, and only those cells will take this particular gene, which will also be expressed in the neuroprogenitor cells. So these are neuron-like, and it will then cause this M. cherry to be expressed. So before you leave, just a couple of things. I'm going to keep the multiple choice test questions from this section really straightforward. You're going to get 3 multiple choice test questions related to knowing something about the opsins and their activation, and the properties of the modified opsins, channel rhodopsin, halorhodopsin, and 1 question on pharmacogenomics, so this last section here, the pharmacogenomics, and then you're going to have 1 short answer question. As I said to many of you who were showing up here earlier, if you can answer all the questions here on this purple sheet, you should be able to get perfect on this section for the long answer. And that's my goal, is to make sure that you understand the material, not just to be able to blindly repeat back to me what I've said in lecture. So I'm going to ask you to do a couple of things. So those questions there, I'm going to ask you to actually share some of your answers on the discussion board. Post your answers. Post what your responses are. I will correct or give suggestions or give you feedback on it. And I'm not going to be taking marks off or anything else, but it's your opportunity to post your responses before you actually do them on the term test. Or if you feel uncomfortable doing that or something isn't clear, come and see me in the office and I can go through that individually with you, provided I have time to be able to do so with you. Okay, so I know we've gone through a lot. On Wednesday, we're going to go into synaptic modification. You'll get the notes posted for that in the next few hours or by tomorrow morning. So please go back and check on that. If you're going to head over to the HMB office to check your term test, please do so. And I'll look to seeing you all on Wednesday.